Hi, this is Miss Litton, and this is my wonderful period 4 class. Say hi.
Hi.
All right, and so we're going to talk about the enzyme lab and the setup for that. So for our enzyme lab, our driving question that I'm giving you is how do abiotic or biotic factors influence the rates of enzymatic reactions? This is going to be an all digital lab, and I'll explain what I mean by that in just a moment. You're doing the lab, you know, you're actually collecting data, but your recording of it is going to be digital. So I'll get to that part, but if you look at the bottom, here is the reaction. Have I already introduced this reaction to you? Okay, so we're going to take hydrogen peroxide, and the enzyme that we're using, this is the name of our enzyme. It's catalase. Okay, and it's going to break it down into water and oxygen. And we already talked about this. Hydrogen peroxide is what color?
Clear.
Clear, and water is clear. But the oxygen will be a gas, and it will what? Bubble. So what we can do is measure the bubbles, okay? And that's what we will use our probeware for. So you'll take your hydrogen peroxide, you'll add in your catalase. It doesn't change colors when it becomes water, but we do have these bubbles. And for your reaction, what you're going to decide as an investigative team is what your independent variable is. Your dependent variable is set. You're going to measure the amount of oxygen, and you're going to measure it in parts per million per second, which you can convert to minutes by dividing by what? 60, okay? You know how to do that, okay? And you will decide what your constants are. You will decide what your control is. You might want to vary what? What's something you could vary? Yes? The amount of hydrogen peroxide. What else could you vary? Amount of H2O2. What else could you vary? Yes? Amount of catalase. Good. What do we know how enzymes work? At a specific temperature. So you could vary the temperature that you do this reaction. They work at a specific what? pH. You could vary the pH. That part is up to you. How you're going to get quantitative data from this, okay, is you will have a reaction chamber, and I have them in the back if you want to look at them, and you will put in hydrogen peroxide. Now, the amount of hydrogen peroxide, regardless of what you do, you could dilute the hydrogen peroxide if you wanted to. It comes as a 3% solution. Or you could take that hydrogen peroxide and you could put it on ice, and you could take its temperature, see what the temperature is. You could change its pH, okay? And you will add in some catalase to it, and oxygen bubbles will form. And this right here is an oxygen sensor, this probeware. It will send the amount of oxygen. Here's this oxygen sensor. We're going to snap it into a Bluetooth who will send it to your iPad, where it will record the data and actually graph it for you and put it in a data table. This is part of the digital doc, is you will be able to send that picture of that graph into your digital document and paste it in there, so you have a copy of that. So you're going to vary, like I said. If you well, I'm going to switch the background. This is what you have to have: the definition of an enzyme, factors that can affect the rate, and the reaction that you're going to be studying. That's maybe 4 sentences. You're going to write your research hypothesis, and if, then, because. This part you will develop. Okay? So let's go back to your lab map that's on the Google Classroom, which all of you need to have one person who shares it with you. Come, lab map. And I put a PDF in Google Classroom, but I also put a link here, but I tried to change that link. For some reason it was saying it was locked. So if somebody could click on that and tell me if it's unlocked or not. If it doesn't unlock from there, it is on Google Classroom as well. Just want to check to make sure you can get it. Had some technical difficulties.
It's on here. Yeah, it's not on here. We don't even have that.
Oh, I put that on there this morning. So if you already did this before this morning, that would be why. You don't have the latest version of it. If you did it like last night, you don't have the latest version when I put it on Google Classroom. So you might want to delete that and reshare it, okay? All right. So let's go over this, okay? If you look up here at the top, you're going to have one submission per team. One submission per team. Whoever opens and shares owns and submits. Okay? Whoever opens and shares own the document, and they're the ones who submits. You're going to do all of your work in what? Blue. And you're going to remove all my prompts before you turn in your final copy. Okay? Before you turn in your final copy. I should see evidence that all members have contributed to the write-up, because all I'm going to do is click on revisionist history, and I will see who you shared it with and what each of you did. I just click on your name so I can see if you're contributing. If you're not contributing to the process, what happens? You don't get the points, right? Okay. You're going to change the title. I have a title right here where it says HBio Lab Map. You're going to change your title to Enzyme Activity period 4, group whatever, and list the last names of your members of your team. Look at me, look at me. You can do that later. Just listen for right now. Steps 1 through 3 need to be completed before you start the lab. And you know how in your handwritten lab notebooks, how I have to sign you off that your pre-lab is ready? What you're going to do here is you're going to do steps 1 through 3, which basically is up through your hypothesis, and you're going to submit that to me on Google Classroom. I will then open it and see what you're going to do, and I will make a comment on there, and that comment is like my what? That's like my signature. So don't resolve it. Leave my comment there, okay? And then once I return it to you, and I'll probably conference with you just to verify what you're doing, once I return it to you, you can start your lab. Now, when you run these, your runs usually last, most students do about 3-minute runs, you know, and you might do 3 3-minute runs or 3 5-minute runs at most. So you will have your control, and then your independent variable, you can do 2 different things, like 2 different pHs or 2 different temperatures, 2 different concentrations, whatever it is that you're going to change, okay? And so if you look at your background, you're going to give me the enzyme overall structure, and I didn't want you to write like a book on that. All you have to do is state that it is a what? Protein, and tell me the functions of enzymes. What is their purpose? Okay, so you can probably do that in 1 or 2 sentences. Your second sentence is the specific metabolic reaction we are investigating. That is, hydrogen peroxide is the substrate. We're using the enzyme what? Catalase to break it down into what? Oxygen and water. Okay? And then factors that can affect enzymes and why they affect that rate. We talked about that in the notebook presentation, yes? And remember, I also had you highlight that portion of your notes? Okay, so that's that part. Then you're going to write your if, then, because hypothesis. I'm going to have you do a research hypothesis, not a null. Do your independent variables, that's what you're choosing, but we're going to measure your dependent variable as the amount of oxygen. What are you going to keep the same? Your substrate is going to be the same amount every time, 5 mils, right? And the catalase, every single time, when you add that in, you will add 1 mil. Okay, so those are things that you're going to keep the same. And you need to look at your experiment to make sure you've addressed all of that. Whatever your control is going to be dependent on what you're studying, what your control is. So you will identify that. At that point, you submit it into Google Classroom. Now, instead of having you draw out like you do in your notebook, your flow chart, this time, once you start doing your experiment and you have your setup, you're going to take pictures of your setup and throw those pictures into this digital document, and that will be your flow chart. Okay, follow? You might need to do some labeling on there, like this is this. You could use a pencil, and a pencil, not a pen, what do I want to say there? Apple pencil to label, yeah? All right. And then you will need to make a data table of whatever you're collecting. Your graph, again, screenshot, boom, throw it in there. You'll have a graph of your data. And it's so cool on these graphs in the Pasco ProbeWare that you will be using, that app on your iPad. You can have all your runs show up just on one graph. And I'll have you practice it a couple times before, obviously, you do the real deal. Okay, then you'll make your claim, your evidence, your reason, and then your conclusion. Okay, now you've never used probably this ProbeWare before, so I have a video that, and oh, by the way, look right here. Read through the entire document first, then read through the rubric. So you know how I'm going to grade it. But to know how the equipment is, I do have it in the back of the room. But there is a video, and you can mock me, I'm good with it. Okay, but I did this while I was holding my phone.
Enzyme activity.
Okay, but when you watch this, it will show you how to.
Catalase will be your enzyme, and the hydrogen peroxide will break down into oxygen and water in your reaction chamber here, and you'll have an oxygen sensor connected here.
Okay, and as you watch this video, I show you how to connect it Bluetooth, which some of you probably intuitively would know, but I'll show you what the software is on the iPad and how you link it to your iPad and how to use and set up a graph and all of it. It's a 4-minute video, so you can see here I'm showing you how to do the link, and I'll show you the app that's on there. Watch that 4-minute video so you know how to do it. Next class period on Tuesday, once you have the comment resolved and I know what you're going to do, then you can start doing the lab and trying the lab out and then do your runs. That will happen on Tuesday. On Thursday, did I already tell you I have to go to a conference? So Thursday I'm at a conference, and you're going to do 2 things. You're going to do your POGL and you're going to finish your group write-up and then submit it that way. And you have an opportunity for extra credit. What's your opportunity for extra credit? Do you know? Yes. When you're doing your lab, at the start, if you take a team selfie, it's number 7 right here, and insert it above your driving question, and it's a good picture, and I can see all of you doing your lab, then you get a +2 on your lab. Yes? So, okay, so you have to decide what your independent variable is, and then you'll be able to write your hypothesis. So, for instance, a hypothesis might be, if I vary the concentration of the hydrogen peroxide, the substrate, then what would happen if you varied the concentration? I don't want to write it for you, that's why I'm not. So you're going to tell me what it is. If I alter the temperature of my enzyme, then and because, what do you think would happen to your reaction if you altered the temperature? If I increase the temperature, or if I what would happen as a result of that, knowing what enzyme activity, how it functions? That's what you have to think through. Okay? And then explain why you think that's going to happen. Okay, what else? Okay? If you choose to work on it over the weekend, I should be able to see, and not one person doing all the work and submitting it, right? You have to all be participants in that and contributing to the process of what you're going to do, or you can talk about it a little bit and then just write it up and submit it right at the start of class, and then you have some chance to work on it. Okay? Questions or concerns? Okay, be smart. Don't stay up too late, and if you do, eat a piece of toast.
It's with some cookie butter.
With some cookie butter?
It's so good.
I haven't ever tried that.